So today, what I really want to talk about is carbohydrates part 2, which is essentially going to talk about polysaccharides. Okay, so some examples of different polysaccharides that we see in nature and how sugars are involved in, for example, cell recognition, how sugars are involved in diseases, that kind of stuff. Okay, so sort of a little, I wouldn't say this is application, but a little downstream of what we talked about. So again, polysaccharides have many different biological functions, particularly for energy storage and structural support. So really, when you're talking about polysaccharide, so long polymers of sugars, really you're talking about either energy storage or structural support. For example, rice we know is full of starch, okay? And starch is made out of amylose and amylopectin. We'll see that in the next slide. Now, trees, for example, your tree trunks, it's very durable, right? This durability is provided also in part by polysaccharides that will form these fibers that's sort of in between the plant cells, that sort of forms this plant cell wall that is going to be used to provide rigidity and strength to your plants. So storage polysaccharides, the principal storage polysaccharides are amylose, amylopectin, okay, regardless of whether you're a plant or you're an animal, okay? But primarily this is talking about plants, okay? Now, so amylose and amylopectin is really the constituents of starch. Okay, so starch is made out of these two things. And glycogen is the sugar storage that's found in animals and microbial cells. For the most part, these things are sort of stored as a granule or a little globular storage that looks like that. Okay, so this is tuber cell granule. So this is all of your amylose or sugar storage. So it's very compact, so you don't occupy a lot of space. Now, glycogen is deposited in the liver, which acts as a central energy storage organ in many animals. Okay, so let's say today, you just had a very good lunch, right? And let's say you eat rice or noodle. That polysaccharide that you eat is primarily amylose and amylopectin, right? You eat it, and then you're chewing it, okay? So you're sort of opening the starch to your saliva, which inside of your saliva, you have enzymes that are going to cut your sugars. Okay, that's going to cut your sugars into smaller pieces, and these smaller pieces, after you digest them, go into your stomach, you're going to cut it into finer pieces, okay, eventually into the monomers. Now, all that sugar you may not need immediately, right? Okay, you don't need it right away. So then your body will have to store it, right? Now, your body does not store it into fats, okay, even though, yes, we know, fat is the energy storage, but fat, lipids is the long-term storage, right? For short-term, what your body will do is it's going to take all these glucose and then assemble them into glycogen, okay? And glycogen has a very similar structure to amylopectin, okay? So essentially glucose polymer. And then inside the liver cell, what you'll see is you see these little thingies, I guess. Those are the glycogen storage. Okay, so they do form a little ball inside of your cell. Okay, and then when your cell, as after you're eating, maybe after 2-3 hours, and you're exercising, and then your cell will take these glycogen polymers and then degrade them and then retrieve the glucose for later usage. Okay, so let's take a look at a little structure. Here, before we show you the structure here, I want to say that amylose, amylopectin, and glycogen, they're all homopolysaccharides of alpha-D-glucopyranose. Okay, now, for the most part, when we say glucose, glucose is going to be pyranose, 6-member ring. That's just normally how it is. Now, the key here is that it's polysaccharide of alpha D-glucose. It's a very specific type of molecule. You're only using D-glucose, and all of that D-glucose have alpha enantiomer. Now, amylose is a linear polymer that's formed by alpha 1,4 linkage, glucose alpha 1,4 glucose. So it's going to be just like polypeptides or DNA, it's going to be a polymer, a long polymer. Okay. Now, in the case of amylose, they actually form a helical structure. Why? Because you have hydrogen bonding between the different residues, okay, or the different subunits. Amylopectin and glycogen, on the other hand, are branched, and glycogen is highly branched because they contain both alpha 1,4 and alpha 1,6. So alpha 1,4 gives you the chain. Okay, it's linear. Alpha 1,4 is linear. And then anytime you have an alpha 1,6, so right here, this guy is an alpha 1,6. This guy is 1, this guy is 6. Okay? Whenever you have an alpha 1,6, you have a branch. And then from here, you can have another alpha 1,4 chain. Okay, so the polymerization, the chain, is always, for amylopectin and glycogen, is always alpha 1,4. But whenever you have an alpha 1,6 linkage, you start have branching. Glycogen is highly branched. Now, let's think about why. Okay, why would you want a molecule branched versus linear? What's the advantage of having branches? What do you guys think? Anyone want to take a guess? Saving space. I wouldn't call it saving space because having branches, you actually occupy more space, right? It's going to be more exposed to the solvent, so it's going to be larger. But it's related to space. Okay? It's a very good direction. Turns out, it has to do with the degradation and the rate of degradation, right? So for sugars, if you want to degrade, okay, we're going to have exo, from the outside, or endo, right, from the inside, just like proteases and just like your DNAs. Okay? Now, if you have a lot of branching, that means that you have more ends that's accessible to the solvent. Okay? That's accessible to enzymes, right? So then, if you're an animal, right, you're exercising, and all of a sudden your cell, your body, is asking for a lot of energy. You don't have time to wait for your enzyme to slowly degrade. You want to very quickly degrade all your glycogen. Okay? So you want to have a fast degradation. Right? Now, having a lot of branches essentially gives you a lot of ends. So you have like right here, right here, right here. So then essentially your enzymes will be more accessible to your substrate. Okay? It's not meant to be a long-term storage. Another thing is that what you see is for sugars, polysaccharides also have directions, just like polypeptides. Okay? Polypeptides have directions. You go from N to C ends, right? N terminal to C terminal. Okay? DNA has direction. You go from 5' end to 3' end. So sugar polymerization. Sugar, of course, you also have a direction. You have the non-reducing end and you have the reducing end. So you should always know the difference between the ends of the sugar. Here we have cellulose. Okay, so this is structural polysaccharides. You have cellulose, which is composed of mainly and only beta 1,4. Okay, so this is still glucose. Cellulose is the most abundant polymer in the biosphere. Okay, it's like everywhere. Trees, for example. Okay, it's like everywhere. It's similar to amylose and glycogen in a sense that cellulose is also a glucan. Okay, so we call them glucans because they're all just composed of glucose. So it's a homopolymer, right? It's made out of only glucose. So only glucose. Okay? Now, different from amylose and amylopectin and glycogen, this guy is a beta 1,4. Now, beta 1,4 linkage, what essentially we'll do is, remember we say that this is sugar, beta points up, right? Okay, so if it points up, the next sugar is 1,4, so this is 1. So the next sugar, 1, 2, 3, 4. 4 is supposed to point down, right? Now, turns out as you're making cellulose, this guy is going to rotate 180 degrees such that you get essentially this picture right here. So this one is the normal glucose that we'll draw. So beta, right? This is beta because it points up, point up. And then here, this glucose is flipped 180 degree, right? Because this guy is C1, C2, C3, C4, and then C5. This is C6. And then C4 points down. That's how it's going to sort of have the bond like there. Okay, so both of them point up or both of them point down. You don't have one point up, one point down. Okay, that's kind of awkward. That's weird. Right? You just don't have that. So what's the reason? You know, we say cellulose. Cellulose, I just told you that it's found in plants and it's used as a structural support. Okay, so most of your trees are cellulose, and then that's what makes it hard. Your paper that you're writing on, it's primarily all cellulose, by the way. Okay, so that's what the paper material is made out of. Okay, so if it's just like amylose, it's glucose polymer, but then just because it went from alpha to beta, how come the property changed so much? Turns out it's because in the beta 1,4 linkage scheme, you end up with a lot of hydrogen bonds, a lot more hydrogen bonds between the different strands, different lines of the polymer. So for example, here we have, this is the oxygen, C1, 2, 3, 4, 4 points down. This is beta, so it points up. What you can see is that you got significant amount of hydrogen bonding between the two strands. So this is one strand, that's another strand. So as a result, they sort of completely get aligned, and then they form very nice hydrogen bonding network, and then you have a very stable complex or a very stable intramolecular interaction. Okay? And then because of that, it's a lot harder than starch. Okay? Now, since we're talking about plants, I'll give you a little background on plant biomass. Plant biomass is composed of primarily three things. You have cellulose that we talked about, and you have another thing called hemicellulose. Now, hemicellulose is just any other sugar polymer that's inside of the structure, and it still forms a fiber. However, instead of all glucose, you can have a mixture of different sugar. For example, you can have glucose attached to xylose, attached to arabinose, attached to mannose, attached to whatever. Okay, so this is sort of a mixed polymer. Okay, so the subunits inside of that polymer, it's not well defined. It does not have a same pattern. It's just has a lot of different sugar inside of that polymer. Now, that's why it's called hemicellulose. Okay, cellulose is all glucose. Hemicellulose, you can have C6 sugars, C5 sugars, and then different C6 sugars and C5s. Another thing that's a lot in plant biomass is lignin. Okay, lignin is what actually provides plants a very robust strength. So as you can see, let's say we take a little cut of the leaf here, you get plant cell walls, and inside the cell walls you get all these little fibers, and then in the fibers you have these cellulose and hemicellulose chains. Now, what link all these chains together is a network of what we call lignin. Lignin is cross-linked aromatic compounds that's super hard to degrade. Cellulose, not too difficult to degrade. There are a lot of enzymes called cellulases that can degrade cellulose and make that into glucose. Lignin, on the other hand, is usually harder to degrade. Why? Because these things are aromatics. Okay, aromatics are pretty stable. Okay, so as a result, the degradation of lignin is usually slower. And it makes sense. Why? Because if your cellulose is very easy to degrade, then your trees are not going to be standing there, right? If you have a lot of bacteria that can degrade cellulose pretty well, and if your plant is only made out of cellulose, then your plant is going to be degraded like almost right away, sitting outside. So you're going to have some lignin mesh that sort of protects the cellulose fibers from degradation by insects or microbes. Okay, so this is actually one of the hardest things to deal with. Right? So as some of you might know, in my lab, we work on engineering of bacteria to produce different things that we want. Either we produce biofuels or we produce useful chemicals. In our study, lignin is something that we always hate because lignin is the type of molecule that locks your cellulose fibers. Okay, so if I only have cellulose, it's not too difficult. Of course, it's still not as fast as if you use just glucose, but cellulose degradation is not too bad. But having lignin in there, it then becomes very challenging. Okay, so for the most part, if you want to use, let's say, tree waste, like tree stuff, what you have to do is essentially use heat, acids and stuff, and then you sort of very violently explode all of the biological components in plant biomass and then expose the sugars. But if you guys are interested, we can talk more about that. All right, so now starting from here, we're just going to talk about different biological structural things that are made out of sugars. One such example is chitin. Chitin is this thing that's used by insect shells or crab shells. Believe it or not, those things are actually carbohydrates. They're pretty hard, as you know. You guys eat crabs. You know that crab shells could be pretty hard, right? So chitin is also a beta-1,4 polymer. However, instead of using glucose, it's going to use this thing called an AG, N-acetylglucosamine. Okay, so glucosamine is going to be this. So I always like it if I look at it like this. So this is glucose. If it has this, then it becomes glucosamine. And then if it has this is the acetyl. It's attached to the N, and then the rest of this thing is glucose. Okay, so that's why this thing is N-acetyl glucose, and then because you have this, it's amine. All right, so NAG. Okay, and it's just like cellulose, it's beta-1,4. So that's why it has a very strong mechanical strength to it, just like cellulose. Okay, but in this case, you also have an acetyl group attached to it. So that was crabs. Now we have for animal support. So the major structural polysaccharides in vertebrates like us are the glycosaminoglycans, or formerly called the mucopolysaccharides, but I prefer glycosaminoglycan. Okay, so essentially they are glycans that has subunits that are glucosamine. Some important examples are chondroitin sulfate, keratin sulfate, and dermatin sulfate. They're found where? They're found in connective tissues, or they're found on skins. So these are the things that are going to provide plasticiness, okay, or plastic nature of your organs. Okay, so they provide not just strength, but they also provide elasticity to your body parts so that you can put things together. So we have already seen N-acetylglucosamine. Now we got some other stuff. You got sugar acids. Okay, so this would be a sugar acid. And you have, this is sulfate. So chondroitin sulfate is made out of sugar acids and this N-acetyl galactose sulfate, galactose amino sulfate. Or keratin sulfate, you know, similar idea. You have galactose plus the same type of monomer right here. And then hyaluronic acid, we talked about hyaluronic acid last time. You have sugar acid attached to your N-acetyl, this is the N-acetyl glucosamine. Okay. So what do you see? Knowing that these three are the three major polymers that are found in our connective tissues, like skin and joint and other stuff, what do you notice? What's common in them? They have charges, okay? Charges can be used to attract water. They can, you know, repel and attract water. So they can attract some hydration there, okay? So that's what makes your skin, you know, has that elastic feature to it. That's what makes your joints be able to compress and then expand, right? If the polymer is made out of just like amylose, amylose has no ability to contract and expand, right? But in this case, all of these materials are able to do that, right? And in part, that's because of the ability for them to attract water and then repel, okay, repel each other. Okay, so for the most part, they're all negatively charged. Acids are negatively charged, sulfates are negatively charged. So because of that feature, they have repulsion among each other. So then you can press them together, you can, but then after you release that pressure, they can sort of regenerate their shape. That's the structural polysaccharides found in vertebrates. Now, also, what do you notice? You notice that you have a lot of, for example, galactose, N-acetylgalactosamine, or N-acetylglucosamine. Okay, so that's why a lot of people are selling the glucosamine as a dietary supplement. Okay, 你们知道那个维骨力就是 N-acetyl， 就是那个 glucosamine 嘛，对不对？葡萄糖胺嘛。Right? 那葡萄糖胺其實 it's not used as 一個 subunit as you can see. Right? 它不是一個 subunit， 它是一個 precursor. Okay? So glucosamine is actually a precursor. So in order for the glucosamine to be functional and incorporated in your body, you still have to have the relevant enzymes to do that. Okay? So just because you take it doesn't mean that, you know, it's going to fix whatever problem you have. So show you a large structure of this structural polysaccharide. What you can see is this is going to have many different things. You have keratin sulfate, chondroitin sulfate, hyaluronic acid. Okay, now in the centerpiece, that's your hyaluronic acid. And again, all of them are polymers. Okay, all of them are polymers without branching, right? But you see all that branching? Let me get there, okay? So you have hyaluronic acid, and hyaluronic acid is going to be attached to these little blue balls that are the link protein. And then the link protein will then be attached to another core protein that will be used to attach your keratin sulfates, which are the long ones, or the keratin sulfates are the small ones, and then the chondroitin sulfates are the long ones. Okay, so what you have here is a sugar and protein conjugate. Okay, so you have proteins attached to sugar, or sugars attached to proteins, however you want to see it. And in your body, you got a lot of these things. Okay, so you have a lot of these things. They can sort of be used as a structural support that provides whatever function that your body needs. Now, Another thing that we're going to talk about is heparin. Heparin is also a glycosaminoglycan. In this case, it's a highly sulfated glycosaminoglycan. Heparin is a natural anticoagulant. Anticoagulants are coagulants 是那个就是那个血会凝在一起嘛。那 anticoagulant 就是那个的抗凝血剂的那种东西啦，它有那个抗凝血的效果。So turns out heparin binds strongly to a blood protein called this thing antiprothrombin 3, okay, and the complex inhibits the enzyme of the clogging process. So this is actually something that is used as a medicine to sort of inhibit clogging in blood vessels. The mechanism, if you're interested, we can talk about that later. Okay, but this is actually sold. Now, glycoproteins, so that's the thing that I wanted to get to. Glycoproteins are proteins that contain oligo or polysaccharide chain on the protein. Now, glycosylation. Glycosylation is the action of putting sugar on protein. We're cool with that? Okay. So glycosylations serve wide purposes, including cell adhesion and recognition. Glycosylation we also just saw in the structural protein, right? We just saw that you have hyaluronic acid, link protein, core protein, and then they attach to different sugars, right? So it's also used for structural, and in many cases, actually in a lot of the cases, glycosylation is used to put sugars on your surface of the cell and then is used as a recognition process. Oligosaccharides can attach to proteins through either O-link or N-link. Now, N-link, you know, is through connection to a nitrogen atom. So this guy is essentially talking about nitrogen, and then this guy is talking about oxygen. Okay, I know it's really stupid. Okay, N-link and O-link. It's just talking about where does the protein attach. Actually, reverse. Where is the sugar attached to the protein? Is it a linkage via nitrogen or is it a linkage via oxygen? Okay, so commonly glycoproteins, the bond between sugar and proteins are formed through either asparagine or threonine. If it's an asparagine, then it's an N-link. This is N-link. If it's threonine, then it's an O-link. So, for example, here you can have N-acetylglucosamine attached to a protein chain. Through attaching to the asparagine that's on the protein chain. For threonine, it's the same thing. Okay, so you can attach different sugars onto proteins at different positions. Okay, now, typically, you form this link at C1 of the sugar. Typically, okay, the anomeric carbon. Cool? We're cool with this? Glycoprotein. Blood types. Blood types. Asian people love to use blood type to, predict your personality, but that's, not scientific at all. But that's not the point. All right, so we have different blood types, and different blood types are referring to different antigens on top of your cells, okay, on top of your blood cells. O type, we say this is the universal donor, right? So you know that if you have O type blood, your blood can be donated to, whoever that needs it. Okay, it can be donated to anybody. Okay, that's why O blood, it's very precious, that's why if you are O blood, hospitals will really like it if you go and donate your blood. Okay, but whatever. A and B blood, they essentially have different antigens. Okay, and these antigens, they differ in the sugar that's attached to the protein that's on the surface of the cell. So O type has 3 sugars. You have a N-acetylgalactosamine. You have attached to a galactose, and then the galactose attached to a fructose. So this is the O-type blood. Now, A-type blood and B-type blood, they have the same thing. So they have the same N-acetylgalactosamine, galactose, fructose. They have the same part. A-type have an extra N-acetylgalactosamine. Okay, so this is an extra N-acetylgalactosamine. The B-type is not N-acetylgalactosamine. It's just galactose. Okay, so A blood and B blood has a different sugar attached at the end of that antigen, while O blood has no sugar. Now, here's a question for you. It would be useful to convert A and B blood to O blood. Okay? Actually, there are companies that are already doing this. They can actually convert whatever blood they collect all into O blood, essentially. Okay? What kind, or how will we do this? Like, how could we do this? What do you think? Develop an enzyme. All you have to do is develop an enzyme that cut this bond, that specifically cut that bond. If you can chop off this bond very efficiently, and then you remove the terminal sugar, then regardless of A blood or B blood that you have, it's all just going to be O blood. So there are companies that are doing this. Okay? So if you're interested, we can talk about that. But however, it's not widely commercialized yet. Why? Because of the efficiency. Let's say you have an enzyme that does this reaction. That's fine. That's fine. But what if you only cut 99%? As long as you have 1% of A blood inside of the sample, you give it to somebody that's a B blood type, they're going to have immunoresponse. They're going to not be okay with it. So currently, it's the efficiency that matters. So it would be great if you can develop an enzyme that's super efficient. But until then, it's safer to stick with the same blood type.